Well, hello everybody, and thank you very much for joining us for this ticks and tick-borne pathogens open community webinar series. My name is Gloria Isabel Giraldo Calderon, and I'm joined here by Omar Harp and with some other staff members from the VEuPath BRC, and we are very glad to have you here. This series of 8 webinars will join by both the VEuPath BRC and the VB BRC, and today we'll focus on ticks and how to analyze omics and field population datasets, specifically in VectorBase. This webinar is divided in 2 parts. So first, the first 30 minutes, I will give you an introduction about the BRCs. I will give some practical demonstrations about VectorBase and briefly tell you how after the webinar, you can contact us, get some help and documentation. During those first 30 minutes, if you have questions, please type them on the chat. Omar, David will be, which are part of VEuPathDB, will be answering your questions. At the end of those 30 minutes, we'll have an open discussion, and you're welcome to turn on your camera if you want and ask your question live or type them. But there's also a web form. So when you sign up, and I'm going to show you the screen at the end, you can also type more questions there if you want. Okay, so first a little bit of introduction. Oh, and remember, this all is being recorded. So if you miss something or you will have to miss the end, you will have the recording to come back to it later. Okay, a little bit of introduction. So the Bioinformatics Resource Centers, or BRCs for infection diseases, is a program from NIH that started in 2004. Initially, there were 8 awards or databases, but over the years, NIH has asked us to merge, and today we are 2 BRCs. So one is the VB BRC, and the other one is VEuPathDB that I represent. And those are the home screen of both of them. And just as a reminder, please, as you come in, please mute your microphone. As I was saying, these are the home screen of both of the BRCs. And the main objective of the BRCs is to provide public access and open access to all these, to our platforms and their tools and their data sets. And here I'm showing you the URLs and links of how to quickly locate the tools that we offer. Again, here, side-by-side page of both BRCs and where you can access all our data, the genomes, and other data sets that we provide. What is the difference between these 2 BRCs? Is the type of organisms that we focus on. So VEuPathDB, we have eukaryotic pathogens, we have vectors, and we have hosts. At VBBRC has bacteria and viruses. And if you want more details
about— Hey, Gloria, can I interrupt quickly? There seem to be some bars that are showing up in the presentation at the top.
I don't know how to disable
this
that.
Might be the live transcript that's causing that issue, potentially.
Well,
let's see if...
Okay, if that I will keep an eye. I thought you were not seeing that. I'm sorry. I thought it was just me.
There we go. So I think
I'll keep an eye on them and,
perfect.
And close them.
Thank you. Thank you, Gloria. And thank you for those who indicated that in the chat that there was that issue. So go ahead, Gloria.
Okay, sorry. That was just me. If you want more details about the NIH BRC program, please go to their website for more details. And okay, but some people ask, what is the relationship between VectorBase and VEuPathDB? So I told you there's 2 BRCs. One of them is VEuPath. And VEuPath, if you've been to our website, this is how the homepage looks like. And at the bottom, we have all these different logos. And those are links to our different sites. We have 11 of them that are organism focused. One of them is VectorBase, and another one is Pyroplasma database. This one has organisms such as Babesia and Theileria. And in another webinar, not today, Omar will be talking to you about that. But today I am going to focus on VectorBase, so one of the sites of VEuPath. The advantage of the VEuPath project is that all of these different organisms that we have, all of these sites, they have the layout of our websites are exactly the same. The only thing that changes is our organism. So if you learn how to use one, you will learn how to use them all. Okay, so what is VectorBase? So VectorBase is the home of vectors of human parasites, viruses, and bacteria. And we host both omics and field collected data. Notice that I have underlined the word vectors is because, as you may have noticed for the little cartoons, in addition to vectors, we also have ectoparasites and things that are not vectors, such as, for example, the bed bugs. Or this snail here, which is an intermediate host. And we also have a species of importance for comparative purposes. We also have the genome of Drosophila melanogaster, or we also have Anopheles mosquitoes that are not vectors of malaria. So we have kissing bugs, we have housefly, stable fly, yellow fever mosquitoes, we have sandflies, we have Pulex mosquitoes, malaria mosquitoes, and as tsetse flies, and of course, we have ticks, and that's what I'm going to be talking about today. This is the URL for VectorBase, and this is the homepage. So in the homepage of VectorBase, here in the header, you will notice that we have a release number and a date. We update the website every 2 months. And that is telling you that last time you updated the website with data or any other improvements to our tool was the 15th of February of this year. Here on the header as well, you can see site search. And here, as you can see in the example, you can type a gene ID or a gene metadata, a symbol, a name, a function. Here in the header as well, you can have a link to your profile. You don't need one to access our sites, but it will be good because it allows you to save your work. Here also we have a link to our YouTube channel where you will find tutorials and later the recording of this webinar. A Reddit where we both BRCs can post, and members of the community, of course, and Facebook and Twitter where we post information useful for you. Something that we show up sometimes here are the banners. And in these banners, we have important information. For example, this TIC webinar that we are having right now, if you don't want to see those banners, you can just close them. At the bottom, as I said, are the links to the other sites of the VEuPath project. And here in the center are these different cards. If you click on each one of these, the image here in the center will change and will give you 1 to 3 easy steps of how to get started with different resources and the tutorials and exercises in PDF format. Also, if you want more of a live communication, you can use the community chat, or you can click here in Contact Us. We're always happy to hear from you. If you're an experienced user or if you never use VectorBase, don't ever think a question is too simple or too naive. We are always eager to hear from you, and I'll help you as much as we can. Notice here this panel is closed. So if I click here in News and Tweets, it will open. So you will hear from us. If you're not much of a social media person, the highlights of our communications are going to be here. Or if you don't have an account on Twitter, here you will see what we post that might be of your interest. Okay, so first of all, we are going to start with where the data comes from. The data that we host is a direct submission from scientists just like you. But we also do literature curation. And one of the purposes of today is for us to hear from you which data about ticks should we include in VectorBase. What do we need? So people sometimes say, oh, I already submitted this to GenBank. I don't want to do another submission. And you don't have to. Actually, you just give us the accession number, for example, from your bio project. And from there, we copy the raw data. If we are talking, for example, about a genome or an RNA-seq. If we are talking about a proteome, we just need an Excel file and we will take it from there. So you don't have to actually manually curate the data. We do it for you. Preferentially, we like to hear from you before your paper publication because it takes us time. So we only update the site every 2 months. So if you said, oh, my paper just got published, can you put this up in VectorBase? It's going to take a while. But if you say to us ahead of time, say, look, I'm working on this manuscript, I have already the data in GenBank, can you please have it in VectorBase? We will not make it public. We'll work behind the scenes. You will be the first person to see your data. And once you let us know, okay, my paper is out, now you can make it public, we will do. So we work closely with you to make sure nothing comes before you want it to. Okay, I have structured this talk in a way in which I'm talking to you first about genomes, transcriptomes, proteomes, and population data. So first, genomes. So the first thing is you will come to data, click on organisms, and notice we have 55 genomes, and this is the list of all the information. But if you're interested in ticks, that's what you type here, and the table gets shorter. You click, and you arrive to this genome page where we have all the metadata related to who published the paper, who is the PI in the project, where are we pulling data from, statistics about the genes, and anything related with the genome. And we have a table of contents on the side to show you, for easier navigation. And here these links are, you can click on them to start exploring or do some data mining about that specific genome. So that's one way to do it, starting from the genome. Another way to do it is to start here inside search and type a keyword. And here you will get different filters on the side. And notice you get from all our species, you are getting hits, but that's not what you want. So for the genes, you only want exotic data, for example. So that's what you type. You select the species that you want, apply the filter. And now in the center panel, you are only seeing tick data. You don't have to select both. You can only select one and apply the filters. And now I only have exotic capillaris, the cell line data. I select my gene of interest, click on it. And again, this is a data set page, but now it's not about the genome, it's about that specific gene. And as you scroll down, notice how I'm moving through the table of contents, and I can open or collapse different sections as I want to explore more data about this gene. Here, for example, I'm looking at the genome browser, and if you click specifically in this link, you will see the genome browser in a dedicated page. We are not going to do that today. I will save that for Omar in our later webinar. You can also start site search with a gene ID. So you know that's Exodus gene IDs, you search for it, and now I only get a card because I was specific. I didn't ask metadata. I use a specific gene ID. I come directly to the page, and again, it's exactly the same page that I just showed you. I can also construct a search strategy from either the search menu, and you can expand them all if it's the first time you ever seen them, to get familiar with what are all the different possibilities that we have. It's also available from here in the home page. But once you kind of know what we have, a good idea will be to start typing keywords. For example, I want genes related to a specific organism. So I click on organisms under genes, and start my in silico experiment or data mining. I reset my values because I have run a search before, and I want exotic data. So I select which species do I want. I only want the reference genome, and I click on Get Answer. You can see I have run a previous search, so I'm going to close that to only show the one that I'm running. In yellow is turning my first step, and is more than 23,000 genes that I'm showing, and that is all the list of genes that I'm seeing here, and these are links to the gene page that I showed you earlier. I can grow this clicking on Add Step, and I can do several things. I can combine, I can transform, and I can do genomic collocation. So I want to combine first with other genes. I can do intersect, I can do a union, 1 - 2, or 2 - 1. I'm going to do an intersect the things in common with a new search. So first I did all the genes of Ixodes. Now I want to know those genes, which one have transmembrane domains. I want to see for the Ixodes genes, I select again my species. And I said, I only want the genes with 7 transmembrane domains. And I click on run step. And these are other data sets for which this query is available. And the program that we use, run step, that's documentation for you if you want more details about this query. So now step number 2 is giving me the intersection between all the genes with transmembrane domains that are in Ixodes. And here, if I click on yellow, so I see again the 23,000 genes that I had initially. But if I go back to step 2 to 180 genes, those become yellow, and those are the ones that I'm seeing here on my list. I can add one more step, and this time I can say, well, I want to see if these are secreted. So I select, probably a signal peptide, again, for Ixodes. I select my species of interest, I run a step, and remember, this is the program that we are using to run this. And I get 10 genes, and those are highlighted in yellow. Those are the ones that I'm seeing here. What can I do? I can copy this strategy, I can edit, I can save, I can share with somebody, I can delete, or I can close, as I showed you before with the other searches that I had open. I can also download this data. So I have selected 10 genes, and those are the download. So I can have a table, I can have FASTA sequences, I can have GFF3 files. For the table, I can customize which fields, which headers do I want. I can see them in browser to know if that's what I want, get genes. And I see this, oh yes, this is what I want. So now that is showing up, so I can actually download in CVS format, for example, and open in a program such as Excel. So that's why I was telling you that it's good for you to have an account, because instead of having all those organisms, you can set your preferences, and you say all the time, I don't want to say Ixodes. Out of the 55 genomes, I'm only using Ixodes. So if you don't want to do that all the time, you can come here and say, okay, clear all the time, only show to me Ixodes, and that's it. You apply your filter, and now notice all the time is only going to show the 2 species that you have selected. And you can enable or disable these at different points when you are exploring the website. So I encourage you to use this if you think you are not but for example, if you want to play with the orthologs from Anopheles or for Drosophila, you may want to disable that thing. These are all my searches, and I have them saved because I have a profile. I can run a blast job, and if I have a profile, I can have them saved. So that's you don't have to, but that's an advantage of having an account. You can save all these preferences in your account. Okay, just to summarize what I've just said, at the top of the website, you are going to have a banner that says My Organism Preferences. You can select how many you want, and at any time, you can enable or disable this feature. When you are running a search, for example, you will not have 55 here. You only have 2 because you have enabled this. And it said you need to select some to run your query. So you can select both, or you can select one, in this case, for example, for Ixodes. So it says you have selected 1 out of the 2 that you have open. Let me see how we're doing on time. Oh, wow. Okay. Other samples, search strategies. I have made all of the ones that I have run, I have made some public for you to take a look. I know I went through this probably very quickly, but you can always come back to this and see what other people have run. And actually in some publications, people publish their strategies as a figure and let people know how they arrive to this. Maybe you don't want to make it public yet, but you want to share with a colleague, you can click here on share, and only the people that you care about will be able to see that strategy. To summarize, so you can come to the tick genomes directly, you're starting from the genome, or you can start from the tools menu. You can start inside search with a gene name or symbol. You can type inside search a gene ID. From the searches, you can start a strategy or an in silico experiment. And I didn't demonstrate how to run actually a blast, but maybe you're familiar with that. Come to the tools, blast, and here is in the help, you will find a 4-minute video that will show you how our implementation of blast works. Which data sets that we have from ticks? Go to data, data sets in VectorBase. And out of the more than 1,000 data sets, you can filter and say, I want to see what data do we have for Ixodes. And now it's showing me I have 29 data sets. But if in different categories, the ESTs, genomic variation, genomes, immunoepitope databases, protein expression, and many RNA-seq data sets, all for Ixodes available for you. So if you're looking, if you want to be more specific, when you come here, you can click in this arrowhead and say, I only want my keyword to be working with the organism column. And that way, for example, look, now I got 26, because maybe before when I type on Ixodes, it's picking up a word for both the summary or the description of the data set, and you have repetitive data sets. So this way, it could be a more refined query for you to try. Okay, now transcriptomes. So you come here to the same data sets page. In category, you type, I only want RNA-seq, or I only want microarrays, any category you want, and now it's only showing you the data sets in that specific category. And you maybe want to say, well, I only want RNA-seq in Ixodes. And see, now we have 22 data sets. And you start scrolling to the table, and you look at the papers, and you select which of the available data sets is of your interest. If you select one of those data sets, again, we'll show you the data set page with all the metadata. Okay, so I have now selected an organism and a category. And this is to remind you, it could be all lowercase or a mix of them, it doesn't matter. The table, it will still work. And the reason why we are able to provide all these RNA-seq data for you, and you can compare them, even though they come from many different assays, is because we pull the raw data and we analyze it under the same pipeline. So we run this through the standard workflow. We compare across all the data sets, we determine the expression value, and we make sure if an author has called a gene as being upregulated or downregulated, is the same as showing up here, even though not maybe with the exact same, TPM. When you select a study, again, the data set page, it will tell you the publication, where it's coming from, and a list of how you can analyze that data. So this RNA-seq, you can do a fold change, you can do a differential expression, a percentile, or a sense or an anti-sense analysis. Sorry. If I'm doing a differential expression, so I'm going to reset my values because I ran a query earlier. My reference samples are going to be the uninfected cells. I'm going to compare them with the infected cells, and which direction I want my comparison. So you can say, I want only upregulated, I only want downregulated, or I want them both. Fold difference, do I want 2, 20, 100? And what statistical significance do I want? And I click to get an answer. Again, those are the queries that I ran earlier. And this just, you can delete them or leave them there. And the one that we just ran are these Ixodes capillaris infected cells. And again, the results are at the bottom. You can give them a name. So you can say, this is any name that you want. This is the one that is going to show up if you decide to make it public. And here you save it for you in your profile. And as I was saying earlier, you can decide to make this public or only for you. In edit, so notice if I hover with the mouse, this edit option shows, you click on it, and you can revise what were the parameters that you selected for your query. And you can change them if you click on revise. You can decide, well, I get way too many genes. 400 is too much. So you can increase your fold change or decrease your p-value. You can download the genes as I showed you earlier. Which data sets do we have for RNA-seq that we can use not only in the genome browser, but for this searches query? So you remember, from both the top or the side, you start typing RNA-seq, and thus you arrive to the page. And here you will get all the RNA-seq evidence for all the species available in VectorBase. But you can filter and say, I only want, which type of search do you want? And you select for Ixodes. So for Ixodes, the one that we were doing before is these cells infected. And we were doing differential expression one because we were interested in getting statistical significance values. And that's the one we did. And here is just showing you again, how to run the query, and exactly for the other ones, for the fold change, which is here, or the percentile, or the sense or anti-sense is again. And Omar will go into more detail in the next webinar when we look at Pyroplasma database, how to set up all these different queries. But today, the take-home message is VectorBase has data, and with the raw data, we process it, and you can ask your own questions, different from the ones that the authors already asked in the papers. Summary. You go to searches, start typing, it suggests a search for you. Once you get to the list, you can hover with the mouse, get a summary, not of the whole paper, but that data set per se, and you get a link to the publication, and you run the type of search that you want. Proteomics. It's basically the same thing. You go to the searches, but you start typing the word protein or proteomes, and you will get to mass spectrometry data. And same, you can do it from here or from here, is the same thing. And once you select the query that you want to run, you set up your parameter. So here, reset the values because I ran one before. I have 17 data sets because, I have not, but these are, so each data set has different samples that I can analyze. And I said, I only want to work with Ixodes. So I can enable here, and it's showing me now I have one data set to select from. How many peptides do I want per sequence? How I want to run the comparison. I can leave the defaults or change them and get an answer. I can also have advanced parameters to look at. And from there, same as we did before with the RNA-seq query for the mass spec, I can just grow my strategy, save them with a colleague if I want to. And I give them a name, save my query. And now that I have saved it, I can share. And with that link, only the person whom you share that link with will be able to use it. And actually, you can also share that link in your publication if you want people to find your query easily. And the last thing before we get to an open discussion is the population data. This is the map in VectorBase. So you go to tools and select the map. And here in the map, we have worldwide data, and we have population abundance, mostly coming from mosquito and other control districts in the US. Is this routine surveillance data in which they have detected which species, which vectors are there, if they are infected with a pathogen or not, insecticide resistance, genotypes and phenotypes, CDC bottle assays, WHO tests, and many different assays. And so if I want to look for tick data, for example, I can just type a keyword here. And I select. So this one, I want to look at that one. So all the points disappear, and I'm only now looking at the data that I selected, and the points start to disaggregate as I get closer to them. So I select a point and now becomes yellow, and it's telling me this is whole genome sequencing, and all the metadata is available here. So this data is coming from Ixodes capillaris. I wonder if there is for more species. I change the filter, I select species, and it's telling me no, all the data is coming from Ixodes. This doesn't mean that we cannot have anything else. It's just what we have currently. So if there were more species, you will have a pie chart distribution. When was the data collected, location, the dataset page, the collection protocol, and the protocol itself to process the sample. So all that information you will have there. You can download the raw data, only matching your search or everything on the map with it, and these are the different columns that will be available in your file. And when you download the data, we remind you to please, as with the omics data set where you take the data from, the original data provider, and then you grab it from VectorBase. And if I click there, so I will get, again, not a genome page, not a gene page, but a data set for a page specifically for this population study. And you can see in the map, so like in the other ones, links to how to interact with the data once you're on the website. That's it. So with that, I will give open the floor to discussion. So remember, this is only the first of a series of 8 webinars. In the one in May, Omar will be sharing with you Pyroplasma database, and we'll share many more things about ViewPath, resources that I didn't have time to go through, more in detail analysis, gene enrichment analysis for RNA-seq, the genome browser, Galaxy, and Apollo. So stay tuned for other webinars. And virus and bacteria as well. Remember, we always want to hear from you, not only today during this webinar, but we always, through all our social media, channels, you can contact us. And if you want to learn more, you can click on help. And we have FAQs, we have virtual events, webinars like the one today. We have workshops, tutorials in PDF, and you can learn all. For example, you're interested, you are using our RNA-seq pipeline. So you want to know what are the parameters, the programs that we are using. If you select webinars, you will have, that's the one we are running today. And if you scroll to the bottom of this page, you will see all the ones that we have run with specific links to our YouTube channel to watch the recordings. And there is where we will put our this webinar, for example. Okay, great. So that's
Thank you. Thank you, Gloria. That was very nice. And I think based on the chats, people were very interested, obviously, in what you were showing. And I think we will continue the discussion. So I'm sharing my screen right now, and we will very quickly switch to the discussion phase. Hopefully, one thing you appreciated from Gloria's presentation is that we can integrate a diverse type of data sets into VectorBase or into any VEuPathDB resource or any of the BRCs. So remember, as Gloria indicated at the beginning, VEuPathDB is one of 2 NIAID, NIH-funded bioinformatic resource center. And VEuPathDB, while it supports vectors of human pathogens, host and parasitic and fungal organisms, the BRC supports the bacterial and viral pathogens, and you will learn more about them throughout this webinar series. And we have a number of webinars which will focus on different aspects of these resources. What I'm showing you here is a strategy that Gloria showed you, and I just want to stress upon you how useful this is as an exploratory tool to mine the data. And as Gloria did, I just ran a search for genes that are upregulated or differentially regulated in a particular experiment, and then looked for ones that also have a secretory signal peptide. So these are genes that are potentially secreted and differentially regulated. But you'll notice I only got 5 genes. The nice thing I wanted to make sure you're aware of is that you can always go back and modify these steps. So I'm just going to edit my first step here, which is a differential expression. I'm going to revise this. And now instead of looking for 10-fold differential regulation, I'm going to change it to 2. Maybe I was too stringent before. And so now your number increases here. But of course, as a consequence, our potentially secreted genes increases as well. And one thing to point out here is that this table is dynamic and allows you to add additional columns, which may help you in understanding what are the functions of the genes within this database. Because every genome that goes through our pipelines, our analysis pipelines, we also map various functional domains to them. And so, for example, here's an example of a gene with an unspecified product. But you can see it also has an interpro domain here, which can tell you something about the potential function of this gene. And again, as Gloria indicated, we'll be highlighting some of these in future webinars. But I'm showing you just, Gloria didn't take you to the genome browser, but this is a gene that she showed. I was actually curious because I noticed as she showed that gene, which has 1, 2, 3, 4, 5 exons. You can see here from the evidence, this is based on real data. We determine where we have evidence for introns, and so each of these lines supports a potential exon base or intron, sorry. And so wherever reads had to be split when they were mapped back to the genome, basically. And as you can see, there's this first exon of this gene. There's some really good evidence here for it. There are about 6,000 reads that support it. The second one also has really good evidence. The third one has really good evidence. There's no evidence for this one, at least from the data that's present. But you'll notice also there's evidence here for potentially additional introns in this gene. And you can see the RNA sequence data down here. There's some nice peaks upstream and downstream of this gene. So again, as a tool to support your research and make sure if you were going to be working on this gene specifically, and you see the RNA sequence evidence in the database, it'll be obviously super important for you to potentially revise this gene model and not solely depend on a potentially automatically generated gene model that may not be accurate. So there were a number of questions in the chat, which I want to make sure we get to some of them. And it is totally, we're more than happy for you to turn on your mic and talk, just not everybody at the same time, so we can actually hear from you. But there were a number of questions and chats in the chat box. Also, I know Gloria introduced David Roos earlier. She mentioned him as being one of the people of VEuPathDB staff. He's actually one of the PIs of the project, along with Marianne McDowell for the NIAID-funded BRC. And he's available as well, and I'm sure we'll have additional things to add as questions come up. But as Gloria mentioned, yeah, go ahead.
No, I was going to say that if people want to speak live, please use the reactions and that raise your hand, so we will be able to, in an orderly fashion, say who's next. But you can just type in the chat.
Good point. So I see Sonia has her hand up. Sonia Bravo, would you like to unmute yourself?
Yes, hello. Thank you very much. Very interesting presentation. I was wondering if the site has any, I see contact us, but is there any site where you have job opportunities? Because we may be looking for someone, and maybe someone that comes into this website may be interested in applying. So is there any way of,
no, that's a good question. We don't have a clearinghouse like that, because that's not part of our mandate from our funders. But we are very excited to help the community figure out what to do, obviously. And One thing we do definitely is we tweet any job opportunities. So if you follow the @VectorBase Twitter handle or the VEuPathDB Twitter handle, also we have a Facebook page if you prefer. We try and post things there on a regular basis, and we definitely would recommend that you follow those.
Okay.
But we don't have a specific page where we keep it up to date with job postings.
Thank you.
Sure. So as other people maybe raise their hands, I'm going up in the chat. I know, David, you've been monitoring the chats too. So obviously, if you feel like, and I'm sure Gloria is looking at them right now, but if you have anything you want to make sure that gets discovered. So one question that came up was whether we have data for soft tick species as well. And the answer is currently we don't. And of course, keeping within the mandate of the VEuPathDB project, as supporting human pathogens and vectors of human pathogens, we also obviously understand that you cannot study these organisms in a bubble, right? And so it's often super important to be able to add additional pathogenic organisms, even if not pathogenic of human pathogens. They may be pathogens of animals, but learning something about what they're doing in the animal may be instrumental to us understanding what's happening in the human host. And so we don't have data for soft ticks right now, but we would love to hear from you in more detail about which specific data sets you were thinking about. And we would, of course, we can put that into consideration for integration. And David, you have your hand up if you would like to go ahead.
Yeah, I just wanted to follow up on Omar's comment there and several other comments in the chat. There have been a number of questions about various genomes and the availability of genomes and why genomes have been published that are not or not yet available in VectorBase. And the short answer to that is that we'd be happy to talk to you or others about data that you may have or may be generating, or data that has been produced by other groups that would be useful for your research. And I think we have a pretty successful track record of working with data providers to get things represented expeditiously. The longer answer is that there are a lot of steps that are involved, and just because something has published doesn't mean that it automatically can find its way into the database. There are always QC issues and data processing issues that can take days or weeks or months, depending on the nature of the data and the problems. So the one-stop shop for any such communications are to click on the Contact Us link on any of our web pages, as Gloria has already shown you. And we'd be happy to respond by email or set up a Zoom call to figure out how we can best support you and the researchers in your lab.
And I'm going to just add to what David said. So we are monitoring many genomes, but you are the experts. So you know what we should have that we don't. So get in touch and let us know what are your priorities.
So I know Sally, I know that was Sally Cutler, that was your question about soft ticks. And so let us know if, obviously, it didn't tell you that, yes, we have the data, but of course, if you contact us, whether by clicking on the Contact Us link, and as Gloria mentioned on the webinar page, we also have a form where you can go in and just add free text information if you want. We would love to hear from you. So somebody asked about whether we have a resource for reservoirs of pathogens. And my answer is that we don't have a resource for reservoirs per se, but we do have a resource for host organisms, and that is to understand how a host responds to pathogen infection. Because as we know, many of the pathogens that infect humans or are transmitted by vectors have intermediate hosts, and learning about how the parasite, what kind of responses the parasites or the pathogens elicit in the host is key to us being able to obviously develop new therapeutics. And so HostDB is one such resource where we integrate host response data. And so as a reservoir could essentially be viewed as another host, And the question is, do we have data for that reservoir that's appropriate for HostDB and can help bolster our knowledge of how a host would respond to a pathogen infection? Gloria?
And not only a reservoir, but if you find a study in which they were looking for, for example, a tick vaccine, and they tested that in guinea pigs, and you think we should definitely have that, guinea pig genome and RNA-seq data set, because you want to explore those peptides that they found that could be potentially be used for a vaccine. So guinea pig, we could load those data sets if you believe that's something that will be useful for the tick community.
So I'm going through the chats again, and of course, anybody else, please feel free to raise your hand, or if you can't find the raise hand button, I know sometimes it's daunting, just unmute yourself and start talking. That's totally fine. We won't be angry. I noticed, let's see, Juan Mosqueda asked about some genomes that are unpublished. I know David mentioned this, but Juan would love to hear from you which specific genomes, and of course, as Gloria mentioned, you're the experts, and so we will definitely rely on you telling us what data sets should be integrated. So feel free, if you want to unmute yourself, you can speak right now, or you can just email us, which is fine as well. Go ahead, Juan. Yeah.
Thank you. Hello, everybody. Can you hear me?
Yes, perfect.
All right. Yeah, there are some genomes that we are very interested in working with, like the Rhipicephalus genomes, like Rhipicephalus microplus and the Rhipicephalus sanguineus. I know that there are these genomes, at least partially, they have been sequenced, and they are in some other databases. So that's why I was wondering, why is it that they are not in VectorBase?
Yeah, I mean, it's a good question. So we do have, so sometimes there's some data sets that are not yet in, at least at a draft stage or completion stage, that allows us to integrate it effectively into the database. So that could be one possibility. Another possibility is just we actually don't know about this particular data set, and we really need to hear about it. So you telling us about it will allow us, and we'll probably contact you directly to give us some more information. And that's obviously very useful. And the other aspect could be, depends on whether these organisms, for example, in this case, we're talking about VectorBase, are these vectors that transmit human disease, which is one of our key mandates. But of course, a vector or a tick that transmits disease to animals would be, of course, a potentially high importance, because you can imagine that these can potentially at some point become pathogens of humans, or we can learn something about their relation. We can learn something from the pathogen that affects animals and apply that to what happens in humans and so forth. So yeah, Gloria.
And something to keep in mind with the ticks is that their genomes are huge. And one of the issues that we have with ticks is that sometimes their assemblies are highly fragmented. And sometimes these assemblies, or there are a few that are to the chromosome level, but they don't have a gene set. So because we handle so many genomes, not only the 55 of VectorBase, but all of the VEuPath, so we have some quality standards. We want the genome not to be as fragmented. We want the genome to have a gene set. And we want the genome to be associated with some RNA-seq on proteome data sets. So people, if we load the genome, people can actually take more advantage of it. So probably there are some genomes that are out there that we have looked into it, but are not quite there yet. And we would love to host everything, but sometimes we have to make these choices.
I know Zachary Kloos had his hand up in case you want to say, and I know David, you have your hand up as well. So
Zachary, did you have a question that you wanted to ask? You're welcome to go ahead.
Yeah, hi. Thank you very much for this presentation. I was just wondering, kind of what the hindrance, like the biggest hurdle to completing the assembly of the Ixodes scapularis genome assembly to the chromosome level is at this moment. I mean, it still consists of like a ton of scaffolds, and I'm wondering what I guess the community could do to potentially complete that assembly to the chromosome level.
Gloria, why don't you comment on that? You would know, you have a more up-to-date view than I do, then I'll add a couple of additional comments.
Yes, well, we are happy to facilitate meetings like this for you to know each other and to start working. But at the very beginning of VectorBase, 16 years ago, we were able to participate actually hands-on with the assembly and the gene set. But again, because of the number of genomes, we're no longer able to do it. So besides facilitating these meetings for you to interact, we cannot help you actually get things done, technically, unfortunately. So that will be actually, the question is all up to you. So let us know, how can we help you to get a group together and you guys, work on improving the assembly, and the gene set. For the gene set, it probably could be easier because you can probably do a liftover from the current Ixodes assembly and move it, or ask NCBI to generate a completely new one. But for the gene set, but for the assembly, it will have to be brand new work or resequencing.
A comment on that and a couple of other points, including responses to questions in the chat. So with respect to the Ixodes genome, as Gloria indicated, right, we are not a sequencing center. Our responsibility is to provide fair access to genomic scale data sets, and we try to expedite and facilitate that as well as best we can. But there are many other people on this call who would have better understanding of the status of the improvement of the Ixodes scapularis genome. My understanding is that there are efforts using new long sequencing technology to provide a better genome, and we will try to make that information available just as soon as it becomes available, which brings me to the second point that I wanted to make, and that is to reiterate this comment about making genome sequences and functional genomics data available. The best way to ensure that happens is to contact us, and to contact us early. If you are in the process, for example, of assembling a high quality tick genomes or improved tick genomes, please do contact us. And it's usually advisable to contact us early. More than 90% of the data sets that we release on VEuPathDB have been provided prior to publication. But and this usually expedites getting data loaded and making sure that it is released at the time that the data providers prefer. But we have a very straightforward data release policy, which is the data providers always own that data. So it is never released until the data provider and we agree that it is properly represented and ready to go live. And the last comment that I wanted to make, there's been a number of questions in the chat about tutorials and workshops. These tutorials and workshops are always announced in banners on VectorBase and all VEuPathDB web pages. So if you are registered as a user, you will receive notification of that. You can sign up for the release notes. But I also want to alert you to what Gloria had already pointed out, which is that under the help menu in VectorBase, we have hundreds of tutorials and links to previous workshops and YouTube videos dealing with all manner of different data and data types and analyses. And since the beauty of this system is that the same platform is used for all of the species we support, you are welcome to explore, for example, metabolic pathways in Plasmodium, or how to identify orthologs in Babesia, and use exactly the same methods to be able to explore similar questions in Ixodes or Anopheles or whatever vector species you may be interested in when the data is available, or now in other species, and infer orthologs of interest by an ortholog transform.
So, and as David was speaking, I went to our workshop page and you can see any upcoming workshops. But if you go to the previous workshops, for example, we had one in December, a virtual workshop, and you can actually take this workshop asynchronously. We have all the workshop material is available here. It's almost like taking a Coursera course without getting the fancy credit if you pay for it. And so, so you can go in here, you can watch the videos and take the, go walk through these step-by-step exercises and learn how to use the resource as a whole. So definitely, I encourage you to do that and take advantage of all the resources we have. And as Gloria mentioned, the Contact Us is a really very useful tool. If you can't find something, it probably means that it's difficult to find. And so contacting us and letting us know and asking us the question helps us, first of all, help you, and secondly, helps us say, well, maybe people are not finding this because it's not visible or something, so it's kind of useful on multiple fronts. I did want to also highlight, and of course, again, if people have questions, please raise your hands. I did show the strategy earlier, and I wanted to make sure we also stress the fact that in VEuPathDB, we leverage homology and orthology across species, across the tree of life, very heavily in the resource. So we have another resource called OrthoMCL, which integrates genomic data, proteomic data, basically proteomes from hundreds of genomes across the tree of life, and generates orthology groups, which we then can, all of the genomes in VEuPathDB get mapped to those. And by that mapping, we can now start making some inferences about whether a gene has a homologue in other vectors, other species of arthropods, for example, or doesn't have orthologs in other organisms. For example, I may want to ask, well, out of these 36 secreted, differentially regulated genes, do any of them have, are any of these unique and do not have orthologs in mammals, for example? And I don't know the answer to this question, so let's go ahead and find out. The answer may be 0, which is not a bad thing, right? It's an experiment we're running here, an in silico experiment. You're not wasting any of your reagents in the lab, just a little bit of time. But I'm going to go down to core data. I'm going to look for mammals. I'm just going to click a couple of times on this. And again, we have lots of webinars that detail how to go through this, which you can find and learn about this. But I'm basically asking for any gene in VectorBase that is in an orthology group that does not contain any mammals. And then I'm asking, I'm intersecting this result with my previous one, which is here in the back, which is these 36 genes. So are basically any of these 36 genes, do any of them belong to the groups that do not include any mammals, right? And so I'm going to run this. And as I mentioned, I don't know the answer. I haven't done this. It's a real experiment we're running right here in the background. And it may take a few seconds, but ultimately you'll get an answer and you'll be able to then take it a step further and analyze it or modify it and so forth. And so we'll let this run for a second that's going in the background, and we'll see if others have additional questions. So Juan is asking, is it possible to run or organize workshops in specific universities? The answer is yes. So we definitely would love to provide presentations directly to your institutes if you like that. We've often hosted what we call virtual lab meetings. So your group or a couple of groups in your department. In fact, you can find, often you'll look in your department, you'll find out, oh, there are people who work on vectors, some people work on fungi, some people work on parasites. And we're more than happy to provide a specific targeted webinar, let's say an hour. And then we also hold multiple virtual workshops throughout the year. And of course, if you're able to pull up a good number of people, we're happy to run something specific up to your region or to your location. And as you can see here in the background, this query ran, and actually turns out 33 of my genes here happen to be unique or do not have orthologs in mammals, right? And so now you can go in there and start exploring these further with that additional knowledge. Gloria?
I just want to say that we reached the top of the hour, and some people it's midnight or it's dinner time. So I just want to say that we are happy to stay here if you have more questions, but if you have to leave right now, remember this is being recorded. Thank you very much. We didn't know what to expect and are very happy that you're here with us, that you had a lot of questions. I'm saving the chat. So if we didn't have time or we didn't reply to you right now, I will be able to later slowly go through them and answer the ones that we didn't. As I said, if you want to stay, please do. And for the ones that have to leave, thank you very much for being here with us. We're officially ending.
Great, thank you, everybody. That was good questions from everybody. Okay, while we're waiting, are there any other questions before we officially sign off? And feel free to unmute yourself if you have a question.
Once you're ready to pause the recording, please let me know or unclose the webinar, Omar, so I make sure I save the very last version of the chat. I've been doing it, but
yes, no problem. I think it also saves it in Zoom, but definitely do it just to have it.
Okay, good.
Yes, there'll be Francine. There'll be a recording, and we will post it to the same page where you registered, so the webinar page. And I'm happy to send an, I can send an email to everybody who attended today, or at least for anybody who registered for today's webinar with a link to it once it's up on YouTube. Give us maybe, I don't know, a couple of days. We have to process it and then upload it, but it should be fairly quickly. Great, thank you everybody for joining. That was wonderful.
Hello, sorry.
Yes, go ahead.
Yes, it was a really nice presentations, and I really enjoyed those presentations. For now, I'm really beginners in chicks on tick-borne diseases. So we are working on really focusing on tick-borne viruses. I'm in Cameroon, so I'm a PhD student. I'm doing the detection of viruses in chicks. And really, I'm really interested about transcriptomics and genomics also. So it was really interested for me to listen to those presentations. And I hope that I could have more information about it in order to, if I want to develop a new project and submit somewhere for financial support. So thank you very much for those informations.
Great, thank you. Thank you, Francine. Go ahead, David.
Thank you for your comments, Francine. I just wanted to point out that we do have a variety of resources supporting analysis of transcriptomic data, either data from others or even data of your own. And there will be additional presentations in this series of webinars on tick-borne viruses. So I urge you to keep tuned to the series of webinars that are planned downstream.
Okay, thank you. I've registered to all the webinars, so I'm sure I will attend all. Thank you very much.
Okay, any other last minute comments or questions from anybody? Perfect. Well, go ahead, Gloria. You can save the chat, and then I'll go ahead and stop the recording.